Okay, our next presenter is AJ Gross. AJ is a rising senior majoring in biochemistry. He spent the summer in Ben Kleinsteiber's lab, and he comes from Princeton, Minnesota. AJ.
Thank you again. My name is AJ. I was in Ben Kleinsteiber's lab, and my project this summer was on improving CRISPR base editing technologies. Okay, so to start, I'm going to give a little bit of background about what genome editing with CRISPR-Cas9 looks like. And so CRISPR-Cas9, as many of you have heard or know, has absolutely revolutionized the field of genome editing. It's made it way more.
Just put it, yeah.
Yeah, okay. It's made it extremely accessible for many labs across the world to use this new technology, and also has decreased the price to where nearly any lab can do it now. It allows for precise insertions, deletions, and point mutations into a genome, and it provides a precise, effective, and permanent treatment. And so there are a few different types of CRISPR technologies, the first of which are the nucleases, and these are what do the insertions and deletions into a genome. But as we look at this pathogenic genetic variants, 50% of this is caused by point mutations in the genome, which this existing CRISPR technology is not well suited for treating. And so this is where we turn to base editing. And so to kind of introduce the concept, base editing is taking this nuclease and adding a deaminase to it, fusing it onto it. And this now allows for precise editing of single nucleotide bases. So as we look back to our little chart here, this blue area for transition point mutations is what ABEs and CBEs are adenine to guanine base editors and cytosine to thymine base editors are able to treat. There is one catch, and that catch is that currently of that 30%, we're only able to target about 25. And so there's a need right now to develop more Cas9 base editing systems so we can unlock this further 75%. This is important because as I'm standing here right now, this very second, base editing is hitting clinical trials for cardiovascular and also sickle cell disease. So what is CRISPR? CRISPR at its core is a bacterial immune system where the bacteria can take foreign DNA and incorporate it into its own DNA in response to pathogenic DNA trying to come into the bacteria. And so you might ask, how does the CRISPR-Cas9 enzyme know not to basically chop up its own DNA? And it does this through the use of a PAM. And so look here, and I suppose it might be easy on this. The PAM is basically a motif that this Cas9 protein will look for within DNA. And so it's 3 to 4 nucleotides long. In this case of wild type, it's an NGG. And this provides a stopping point for the Cas9 to then unwind DNA and check for how complementary it is to a given guide. So this PAM is really important when we think about genome editing, because wherever this PAM is the only place that we can actually go in and make edits. So in the case of wild type, an NGG PAM is required. And when we're thinking about using wild type base editors in clinics, you cannot do any sort of genome editing if you don't have this PAM. So in this example of the figure up here, you can see that region in the middle does not have a PAM, and therefore the Cas9 cannot make any edits. But the cool thing is that we can actually engineer new Cas enzymes to target new PAMs and unlock new target sites. In the context of base editing, there's also one more limitation, and that's the base editing window. And this is basically how far this deaminase can reach off of the Cas9 and make edits to nucleotide bases. And so in the case of my example up here, I edit windows between 10 and 20 bases. And so to take an example here, if we have a genetic mutation that we need to target, if we go and try to use wild type SB-Cas9, it's actually unreachable in many cases. That's that 75% that I showed earlier. But what we can do is engineer new Cas9 variants that have an NGA PAM, for example, that can position the edit window in such a way where we can actually go through and treat this genetic mutation. So you might be asking, how do we create new Cas9 variants? And this starts with identifying them. And so the bit of methodology here in the Kleinsteiger lab starts with mutagenesis of the Cas9 and more specifically the PAM interacting domain, this PID. And by changing amino acids at certain positions, we can actually adjust the PAM specificity of a Cas9 and make it target new PAMs other than the wild type NGG. From there, we do some selections and validation steps where we transfect, lyse, and harvest. We then go through and sequence our new Cas9 variants, and we end up with something that looks like this. And so this is actual data that I used to then make my new base editors for the summer. But what we do with this is we go through and look, it's super easy. You just look at what has the highest percent modifieds. These are your good Cas9 variants. And so we choose these good ones and create new base editors. And this is where I picked up during the summer. So from here, what my workflow was transfecting our new base editors into human cells, harvesting the DNA, and then performing next generation sequencing on them. And by doing so, you get something that looks like this. And so this is one of my candidate enzymes. The PAM that it recognizes is NGA, and that's what it's meant to work best on. On the y-axis, you can see the percent base editing is rather high, which I'm quite proud of. But also, you can see on the x-axis the adenines that we're able to edit in the positions 4 and 7. And so this is specified by the guide. I didn't only do one guide for all my enzymes. I actually went through and did 6. So 4 NGA guides to guide our Cas9 and check for complementarism at NGAs, and then also 2 NGC guides. And so you might look at this and be like, well, this is an NGA candidate. Why is it working on NGC? And so this is actually more of a non-specific variant, unfortunately. While I do get very high editing, it's not very specific, which might be what you would want in a clinical setting. Instead, you might want something that looks more like this. So this is an NGC candidate of mine. This variant was a CBE, so editing cytosines and the thymines. And so you can see very good editing on our NGCs here. Not so much on most of our NGAs, with the exception of this one. And so throughout the summer, I did this on a total of 12 enzymes and 12 guides. You can see various different efficiencies using each enzyme candidate here. And so in conclusion, my summer project here in the Kleinsteiger lab was creating new base editing Cas9 variants that can target areas that are previously untargetable. So going back to our little chart here, where we only have wild type, and we're only able to target 25% of genetic point mutations. What we do here in the Kleinsteiger lab and what I have done over the summer is unlock more of this. And so this is more like 66% of genetic mutations that we can now target using base editing. And so my future work within the Kleinsteiger lab will be about testing more of these Cas9 variants, cataloging them, testing their efficiencies on all PAM sites, as well as the ones that they do work well on and do not work well on. And so my acknowledgements, obviously Ben, my PI, for being such a great mentor this summer and really helping me out, not only in the lab, but also with some great career advice. Logan for teaching me pretty much all of this and everything I know now within the field of genome editing. But I'd also like to thank Chris, Emily, Rachel, Joanna, Connor, Lenuan, Bonnie, Jake, and Blair, all for helping answer dumb questions I've had or just help out around the lab. And then also thank you to Sue for putting on such an amazing program. Do we have any questions?
For AJ. I see a hand.
Oh.
Okay, great.
Thank you all.